I am Dr. Sherry-Ann Brown with Fits on the Go, and I'm here with Dr. Professor Ray. Professor Ray, welcome to Fits on the Go.
Thank you very much.
So we'd like to talk with you about a couple of things today. We're really excited about the trial that you'll be presenting. And why don't you tell us a little bit about how the technology works?
RNA interference is a really exciting technology. And what it gives us the opportunity to do is actually to target and silence specific genes, i.e. by targeting the messenger RNA. The second important thing about this is actually it's part of the body's own intrinsic process for silencing the effects of those genes. When we say silencing, we're not talking about permanent silencing, we're talking about attenuating the effects at the molecular level. So how does this work? Well, essentially, people are aware of so-called antisense technology. So these are oligonucleotides. So you've got 22 base pairs, which you inject, which are then taken up by the target organ tissue, and the oligonucleotides, the antisense, binds to the gene of interest and the messenger RNA, and then initiates breakdown of that messenger RNA, so the protein or the transcript is not produced. So small interfering RNA or siRNA is an intrinsic process. Now what happens here is that you have, instead of a single-stranded RNA molecule, you have a double-stranded molecule. And this is an intrinsic process already present, so what we can artificially do is manufacture those oligonucleotides. And so these are again about 22 to 23 base pairs, there's an overhang, but the technology is really interesting because normally whilst this little oligonucleotide, double-strand oligonucleotide, can't get into the cell, it's actually just filtered and excreted by the kidney, so it doesn't stay in the circulation very long, and it goes out. So how do you do that? Well, you can do that with chemical modification. You can stabilize it. And then if you think about delivery, what's the challenge that we next have? How do I get it there? So we've always tended to use large volumes, right? So here now, by modification of the oligonucleotide, by putting a compound called GalNAc on the end, there's a specific liver receptor that picks up proteins that are, getting older, senescent, and the specific bits that the liver receptor looks at is GalNAc. So now it's on this oligonucleotide. So when you inject this compound and you've stabilized it through chemical modification, so RNA nucleases that circulate will just destroy this and break it down, it gives it that longer half-life, right? And it's picked up by the liver. So when you inject it, it all goes to the liver. So this is a great way of targeting hepatic genes.
Thank you so much for being with us. This has been Fits on the Go with Professor Ray. For more videos, visit us at youtube.com/fitsonthego.